what is important to us is to understand how they can specifically regulate one of their downstream pathways, so axon pathfinding and retinal axon pathfinding, and without affecting the other many cellular processes that they regulate. So we're going to talk about cyclic AMP, cyclic GMP, and calcium today, and mostly focus on what they do and how they do it in the visual system. So I'm going to talk about axon guidance and about two axon guidance molecules, or axon guidance molecule family, ephrins. So I'm pretty sure you've heard about them. They're important for retinal topography, and that means that two neighboring retinal ganglion cells that are in neighboring areas in the retina will arise in neighboring areas in the superior colliculus and the DLGF. And this topography is regulated by a gradient of repellent molecules, ephrins, that are expressed in an anterior-posterior gradient in the superior colliculus, together with a gradient of their receptor, FAs, in the retina. So there are other things that are important for retinal topography. So ask Michael, he will describe that way better than I would do. So, but for now, we're going to talk about ephrins today and about slits. So slits are another family of repellent guidance molecules that are important at the chiasm, and they prevent retinal axons to escape from their path. So we have here two repellent molecules, and we're going to try to understand how second messenger can regulate their response to these two guidance molecules families. This story started quite a long time ago, focusing on cyclic AMP. So one of these second messenger I introduced a few minutes ago. So cyclic AMP is important for axon pathfinding, but it's important for many downstream pathways, many cellular processes. And if you focus only on developing axons, it is important to regulate axonogenesis. So the axon, this neurite that will become an axon, is actually a neurite with a high concentration of cyclic AMP and a low concentration of cyclic GMP. And the dendrites, or the neurons that will be dendrites, have a low concentration of cyclic AMP, high concentration of cyclic GMP. Later on during development, cyclic AMP is important for attraction and axon guidance. So the activity of PKA, one of the downstream effectors of cyclic AMP, is required for the attraction of netrin 1. So a netrin 1 gradient is able to attract developing spinal neurons, developing spinal axons, if PKA is active, but if a repellent axon guidance molecule PKA is inactive. If you check a bit later and focusing this one on ephrins, a specific enzyme that synthesizes cyclic AMP is called adenylyl cyclase 1. There are 10 adenylyl cyclases, but if you remove just one of them, adenylyl cyclase 1, their repulsion induced by ephrin A5, that you can see here on this axon, is prevented in axons that lack adenylyl cyclase 1. And that translates in vivo into an exuberance in the terminal arbors of retinal ganglion cell axons in the LGN and in the superior colliculus. So without adenylyl cyclase 1, you have these exuberant branches that are present until adulthood. And with this observation, we were wondering why removing only adenylyl cyclase 1 was sufficient to induce a phenotype. So basically, the idea would be that there are other adenylyl cyclases that are expressed in retinal ganglion cells, but removing just one of them is sufficient to prevent the cyclic AMP signals that are important for the wiring of retinal axons. One of the phenotypes induced by the lack of adenylyl cyclase 1 is the intermingling of ipsilateral in red and contralateral territories in the DLGN. So when adenylyl cyclase 1 is missing, territories that are not intermingled in wild types or merged in adenylyl cyclase 1 deficient mice. So we wanted to know whether adenylyl cyclase 1 would be the only adenylyl cyclase important for this process, or whether removing other adenylyl cyclases would be sufficient to induce a phenotype as well. And when we removed adenylyl cyclase 5 or adenylyl cyclase 8, there was no phenotype at all in the animal. So suggesting that the adenylyl cyclase 1 dependent cyclic AMP signals have a specific features that enable them to control the wiring of retinal axons. That was confirmed when Jeff Goldberg actually showed that soluble adenylyl cyclase 10, controls the outgrowth of retinal ganglion axons, and that adenylyl cyclase 1 and adenylyl cyclase 8 don't control this outgrowth. So if I summarize this question, we have many adenylyl cyclases that all synthesize the exact same molecule, not a variant of the same, the exact same molecule with the same chemical structure, cyclic AMP, and that cyclic AMP then diverged onto the regulation of many downstream cellular processes. And the question we ask is how one adenylyl cyclase can generate a signal with specific features, and how these specific features would control only the guidance and targeting of retinal ganglion axons. And we started studying that quite a few years ago, focusing on the compartmentalization of the second messenger. The second messenger are diffusible in aqueous buffers, but in cells and in the cytoplasm, they don't diffuse that much. And we focused on different compartments. So one of them being the lipid rafts. Lipid rafts are compartments of the plasma membrane. They are enriched in single lipids and cholesterol. And using genetically encoded biosensor, we're able to detect the signals located in lipid rafts or the signals located outside lipid rafts. In lipid rafts, we found that ephrins 5 induced a decrease in cyclic AMP, whereas outside lipid rafts, we didn't find this decrease. To check whether this cyclic AMP signal, this local cyclic AMP signal restricted to lipid rafts, was sufficient to modulate the response to ephrins, we used a guidance assay, a repulsion assay in vitro, and we added ephrins 5 on growing axons. So the control axon expressing GFP retract when exposed to ephrins 5. When we use a cyclic AMP buffer that is genetically encoded, and since it's genetically encoded, we can add a few targeting sequences to restrict its expression in lipid rafts or outside lipid rafts. And only when cyclic AMP sponge, this cyclic AMP scavenger, is targeted to lipid rafts, it prevents the axon retraction induced by ephrins 5. When cyclic AMP is buffered outside lipid rafts, the axon retracts just fine. So that suggests that we have in lipid rafts an ephrins-dependent signal that modulates the concentration of cyclic AMP, and that is important for axon retraction and axon pruning. So we checked in vivo. I'm not going to detail that further because it's published, but the underlying conclusion here is that in the non-drug fraction, we might have as well cyclic AMP signals, but it is important for other cyclic AMP dependent processes, not for axon guidance, or at least not for ephrins dependent axon guidance. And while building this model, we came up with two additional questions. The first one would be, what about other second messengers? So what about calcium? What about cyclic GMP? And not only about the compartmentalization of the second messenger, but also we're interested also into their interaction. So this second messenger are highly interdependent. So we wanted to determine how they influence each other during axon pathfinding. This first study focused on ephrins, and the second question we had was about other axon guidance molecules. And the underlying question is, are these second messenger signals the place or the signals that integrate the different exposure to axon guidance cues? So have we here identify a signal that add up all the signals generated from axon guidance molecules? And that was actually our hypothesis. I'm just gonna give you a hint here that that's not at all what we found. So the first question we had when designing this more general project was to find a way to manipulate cyclic GMP and calcium with subcellular resolution. And when we started the project, such tools didn't exist. So we started with generating tools enabling to manipulate cyclic GMP and calcium in epidraft or outside epidraft. And that's when we started generating a tool called SPONGI, for sponge, inhibiting cyclic GMP signaling. That's the work of Oriol, a previous postdoc in the lab. He's now in Barcelona, and together with Sandrine Couvet, an engineer in the lab, was very helpful with the generation of the many constructs that we needed for this project. So SPONGI, this cyclic GMP scavenger, is genetically encoded, so we could later on target it to subcellular compartments. And it's mostly a piece of PKG. The chimeric PKG between 1 alpha and 1 beta to increase the affinity for cyclic GMP, but mostly it is the binding site of PKG, the cyclic GMP binding site of PKG. But SPONGI lacks the effector domains of PKG, meaning that you can see that as a dominant negative form of PKG, preventing the activation of cyclic GMP downstream pathways. We measured the affinity of SPONGI for PKG, it's around 1 micromolar. It doesn't say much, it's in a cuvette, so it doesn't help much understanding what it does. So we wanted to know what it does into cells, into living cells. And we co-express SPONGI with a biosensor enabling to monitor cyclic GMP concentration. The cyclic GMP concentration here is color-coded from blue, low cyclic GMP concentration, to yellow-red, higher cyclic GMP concentration. When we add nitric oxide onto cells that express only the biosensor, the cell switch from blue to this greenish, yellowish, reddish color, highlighting a higher concentration of cyclic GMP. But when SPONGI is co-expressed, these cells do not exhibit this elevation of cyclic GMP, and that is what is illustrated here on these traces as well. Meaning that we do have, with SPONGI, a cyclic GMP scavenger that works in living cells. Is it sufficient to prevent the deactivation of downstream pathways? We use VASP as a proxy. VASP is phosphorylated when the guanylyl cyclases produce cyclic GMP. So we increase cyclic GMP, detected an elevation of the VASP phosphorylation, but when SPONGI is co-expressed in the cell, this elevation of VASP phosphorylation is largely reduced. So we do have, with SPONGI, a scavenger that is sufficient to prevent the downstream effector. Cyclic GMP is quite close to cyclic AMP in terms of chemical structure, so it was important to check for the specificity of spongy for cyclic GMP over cyclic AMP. And that's why we use a different biosensor monitoring this time cyclic AMP. So with spongy here or without spongy here, the cells respond exactly the same way with an elevation in the signals detected by the biosensor, meaning that spongy is specific for cyclic GMP over cyclic AMP. All of that was in vitro, and we wanted to have something that was usable in vivo, so we electroporated spongy in the developing cortex at E14, and we check at E18. The RFP electroporated animals exhibit a nice lamina of electroporated neurons at the cortical plate. In contrast, when spongy was expressed, we have neurons that are slower in their migration, together with neurons that form heterotopia here and here at the surface of the cortex. These heterotopias are mostly reduced at P15 a bit later, but some of them are still present, and we detected as well at P15 abnormalities in the orientation of the apical dendrites of these neurons, of the electroporated neurons. So we do have with spongy something that can that is useful in vivo. Still, if you remember why we started developing spongy, it was because we wanted to have a way to manipulate cyclic GMP with subcellular resolution. So we fused spongy to different targeting sequences to restrict its expression to subcellular compartments. And we used a lean sequence derived from lean kinase, which is kinase that is located to lipid rafts. So lean spongy is targeted to lipid rafts, or at least that's what we hope. And spongy KRAS is targeted outside lipid rafts, but still at the plasma membrane. So they are found at the plasma membrane, both lean spongy and spongy KRAS. And we used membrane fractionation to evaluate whether or not they would be reduced, they would be confined in the compartment we hope they would be confined in. Lean spongy, after fractionation on a spot gradient, was expressed in fractions 3 and 4 of this fractionated plasma membrane. And that's where we found caveolin as well. Caveolin is the protein that is found in the graft. Spongy KRAS is more found in the higher density fraction, like adaptin, which is a protein, a marker of non-RAS plasma membrane. So we have spongy in lipid raft, spongy KRAS outside lipid raft. And to check whether it would be active or do different things when targeted in or outside lipid raft, we express either the soluble variant of spongy, so without targeting, the lipid raft targeted spongy, or the lipid raft excluded spongy into retinal cell axons. And we expose these axons to ephrins 5. When exposed to ephrins 5, the growth can collapse, so it loses this nice fan shape. And that's a proxy of efficient repulsion by ephrins 5. When spongy is expressed, or when lean spongy is expressed, so spongy in lipid raft, its collapse is reduced, but not when spongy KRAS is expressed. So we do have a way with lean spongy and spongy KRAS to act differentially on different subcellular cyclic GMP signals. So that is about cyclic GMP. I announced as well that we were interested in calcium. So we ran a similar project, but trying to develop a calcium scavenger to manipulate calcium with subcellular resolution. That is the work of Ross again, together with Sarah Baudet. Sarah is a PhD student in the lab, and with the help of Fiona Roche, who's an engineer. So I'm going to go a bit faster for calcium. The design of the project is exactly the same as the one that we use for spongy, except that here we call this tool SPICY for sponge, inhibiting calcium, like calcium signaling. SPICY has two high affinity sites for calcium deriving from valbumin and two low affinity sites from calmodulin. Once again, the effector domains are removed. So we co-express SPICY with the calcium biosensor, induce an elevation of calcium by dumping dasigardine onto these cells, and recorded the change in the calcium concentration. And this change is the blue trace in control and the orange trace in the SPICY cell. So there is a reduction in this elevation of calcium induced by the expression of SPICY. Like sponges, SPICY is sufficient to prevent the phosphorylation of the downstream effector, and here it's ERK. So ERK is phosphorylated downstream of dasigardine stimulation. And this phosphorylation is reduced when SPICY is expressed. It's not as efficient as BAFTA, this line here, but still the phosphorylation of ERK. Does it work in vivo? Yes, it does, and we use the same approach as for spongy. And at E18, we found many of these volcano-shaped neuronal migration streams here that at P15 translate into massive heterotopia sitting on top of the cortex. So SPICY works in vivo. It can be targeted to lipid rafts or outside lipid rafts using the same domains, the same targeting sequences that we use for spongy. And like spongy, it prevents the collapse of the growth cone induced by epphrin A5 when SPICY is not targeted, or when SPICY is targeted to lipid rafts, but not when SPICY is excluded from lipid rafts. So here I would just give you a few examples of the targeting that we achieved using SPICY here. So we targeted specifically the primary cilium. We use NLS and NES sequences to target SPICY to the nucleus or to exclude it from the nucleus, and all of that was efficient. So we do have with spongy SPICY a way to control the concentration, or manipulate the concentration of cyclic GMP and calcium in living cells and in subcellular compartments of living cells. So let's go back to what I announced at the beginning of this talk. So what about the cyclic GMP and calcium signals downstream of ephrins A? Are they compartmentalized or not? And what about the compartmentalization of the second messenger downstream of other guidance molecules? And that is the PhD project of Sarah Baudet, who again has been helped in this project by Fiona Roche. So what Sarah did to monitor first the second messenger signals in developing axons was to use a cyclic GMP biosensor. It's called THPD5BV, but I think we're going to keep calling it the cyclic GMP biosensor. It's going to be easier for everyone, I guess. And we tried to target this biosensor to lipid rafts or outside lipid rafts. But unfortunately, the signal to noise ratio was quite awful. So we used a different approach. We used a soluble variant of this biosensor, and we exposed the retinal axons to ephrins. And in these retinal axons, we co-express the lipid raft targeted, the soluble, or the lipid raft excluded spongy. So what is recorded when the lipid raft targeted spongy is expressed is not the signal derived from lipid raft or generated in lipid raft, but also remaining signal. And this way, we've been able to detect where the cyclic GMP signals induced by ephren A5 originate in the cell. So now what did we detect? So Sarah expressed this biosensor in retinal ganglion cell axons, exposed the retinal ganglion cell axons to ephren A5, and found that there is an elevation of cyclic GMP induced by ephren A5. This elevation is prevented by the expression of spongy, not targeted everywhere in the cell. It is prevented as well by the expression of lean spongy, so targeted to lipid raft, but it's not affected by the expression of spongy KRAS, meaning that the origin of the cyclic GMP signal that is generated downstream of ephren A5 is in lipid raft, exactly like what we found for cyclic AMP. What about calcium? We used a different biosensor, a calcium biosensor. It's called Twitch2B. And Twitch2B was good enough to be targeted to lipid raft or outside lipid raft and record directly the calcium signals. And what we found is that upon ephren A5 application, the calcium signals that was detected was an increase in the frequency of calcium transient. So that's illustrated here using a cytoplasmic variant of Twitch2B. And that increase in the frequency of calcium transient was found again when using a biosensor targeted to lipid raft. But it was not found when using a biosensor targeted outside lipid raft. So once again, downstream of ephren A5, the calcium signals are restricted to lipid raft. All these cyclic GMP and calcium signals important for axon pathfinding. We go back to similar experiments I've been showing you earlier. Expressing spongy, either the cytoplasmic variant or the lipid raft targeted variant or the lipid raft excluded variant, and exposing these retinal ganglion cell axons to ephren A5. And the collapse induced by ephren A5 is prevented by spongy everywhere in the cell, by spongy in lipid raft, but not by spongy outside lipid raft. So that means that the cyclic GMP signals that are important for the cross-cone collapse induced by ephren A5 is located in lipid raft. And this is the one that we recorded with our biosensor. Similarly, the calcium signals important for the collapse of the growth cone downstream of ephren A5 are located in lipid raft because the lean spicy is sufficient to prevent this collapse, but not spicy KRAS. So preventing calcium signaling in lipid raft is sufficient to prevent the growth cone collapse, but preventing calcium signaling outside lipid raft doesn't affect the collapse. So if I summarize what we found for ephren A5, we have one compartment, lipid raft, that groups all the signals that are important, or at least the ones that we detected here. So a decrease in cyclic AMP, an increase in cyclic GMP, and an increase in the calcium transient rate. That doesn't mean that there is no cyclic GMP, calcium, or cyclic AMP outside lipid raft. It just means that these signals are not important for the response to ephren A5. What about slits? Our hypothesis here was that it would be exactly the same. But surprisingly, that's not at all what we found. So we used here a cyclic AMP biosensor, and we detected, like for ephrine, a decrease in cyclic AMP when we use a soluble variant of the biosensor. When we use the biosensor targeted to lipid graph, we didn't detect any change in the cyclic AMP signal, but we found it back when using a biosensor targeted outside lipid graph. So this is exactly the opposite that what we found for ephrine, meaning that for SLIT, the cyclic AMP signal is located outside lipid graph, not inside. We use the same approach to verify that the signal detected is the one that is important for axon pathfinding. We use the collapse assay again, and the cytoplasmic variant of cyclic AMP sponge, the cyclic AMP scavenger, is sufficient to prevent the collapse induced by this one. The lipid graph excluded variant is sufficient as well to prevent the collapse, but not the lipid graph targeted variant of cyclic AMP sponge. So confirming that the cyclic AMP signal regulating SLIT response is outside lipid graph. We reproduce the same approach for cyclic GMP, and sure enough, we found that axons exposed to SLIT1 have this elevation of cyclic GMP when we use a soluble cyclic GMP biosensor. This elevation was prevented by the blockade of cyclic GMP, buffering of cyclic GMP outside lipid graph, but was not affected by the buffering of cyclic GMP in lipid graph, suggesting that the signal that is important or that is generated by SLIT1 is originated in or outside lipid graph, not in lipid graph. To confirm that it is important for axon pathfinding and for the collapse induced by SLIT1, we used again the same approach. We express spongy, either the non-targeted variant or the lipid graph excluded or the lipid graph targeted variant. And sure enough, the cytoplasmic variant and the lipid graph excluded variants are sufficient to prevent the SLIT1-induced cross-cone collapse, not the variant of spongy that is restricted to lipid graph. For calcium, it's exactly the same story. We again use a soluble sensor, lipid graph targeted sensor, or lipid graph excluded sensor. And the soluble sensor detected downstream of SLIT1 an elevation of calcium transient frequency. So that's the entire cell. This increase in the frequency of calcium transients was not affected, was not detected by lean twitch 2B, so the sensor targeted to lipid graph, but it was detected outside lipid graph. So once again, the signal downstream SLIT or outside lipid graph, not inside. And preventing calcium signals outside lipid graph is sufficient to prevent the cross-cone collapse, and altering the calcium signal in lipid graph didn't affect the cross-cone collapse. So as a summary, we have similar signals in lipid graph for ephrine 5, outside lipid graph for SLIT1. And in both cases, it is a decrease in cyclic AMP, an increase in cyclic GMP, and an elevation of the frequency of calcium transients. I told you that we were interested in as well in understanding how the second messenger interacts, and to determine whether this interaction might increase the different signals that are available to the cell to increase the diversity of the regulation of the downstream pathway. So we combined the expression of one biosensor, let's say a cyclic AMP biosensor, with the buffering of another second messenger, let's say spongy buffering of cyclic GMP. And that way, we have a way to check whether cyclic GMP signaling affects cyclic AMP signal. So that's what we did, and that's what we did first for ephrins. In monitoring cyclic AMP, there is a decrease in cyclic AMP upon ephrins addition, and this decrease is not affected by the buffering of cyclic GMP or by the buffering of calcium, meaning that cyclic AMP is not affected by these two second. We monitored cyclic GMP, and we detected an elevation of cyclic GMP downstream of ephrins that was reduced when cyclic AMP was buffered, but not affected when calcium was buffered. So there is an influence of cyclic AMP on the cyclic GMP signal. Similarly, we monitored calcium, found that there was an increase in the calcium transient frequency induced by the ephrins that was blocked by the expression of the cyclic AMP buffer, but not by the expression of the cyclic GMP buffer. So just to summarize that, in lipid rafts downstream of ephrins, there is a decrease of cyclic AMP that is important for both the increase of cyclic AMP and the increase of the calcium transient frequency. And all of that is important for axon retraction. What about FIT? We used the same approach and found that the decrease in cyclic AMP downstream of SLIT1 was blocked by the buffering of spongy, but not of cyclic GMP, but not by the buffering of calcium. Cyclic GMP signals are affected by calcium buffering, but not by cyclic AMP buffering. And the calcium signals are affected by cyclic AMP buffering and by the cyclic GMP buffering. So let's make that clearer in a small scheme. We have now two different compartments with two different second messenger interactions. One, which is generated by ephrins, the decrease in cyclic AMP upstream of an increase of cyclic GMP and an increase of the calcium transient frequency. And outside lipid raft, a different second messenger interaction that is induced by SLIT1. So this network of second messenger in this case is a decrease of cyclic AMP that is upstream of a calcium transient frequency increase. And there is a two-way interaction between cyclic GMP and calcium. Similarly, cyclic GMP influences the cyclic AMP. So overall, we have two compartments and two networks. So that is all in vitro, and these are experiments that we couldn't perform in vivo. But we wanted to validate that what we detected was true in vivo as well. And we expressed our construct targeted to lipid raft or outside lipid raft into retinal ganglion cells, and used the light sheet microscopy to reconstruct the axons of retinal ganglion cells. The controls here, this is the superior colliculus that you see. So the controls have a lot of different terminal arbors that have no axon escaping these terminal arbors. When lean spicy, so blocking calcium in lipid raft is expressed in retinal ganglion cell axons. There are axons that escape these terminal arbors, and we even find axons that go into the inferior colliculus, which is not a retinal ganglion cell axon target. And this is completely in line with the lipid raft localized signals that are important for the response to ephrins, which are guidance molecules important for the topographic mapping in the superior. Instead, spicy so buffering calcium outside lipid raft, the animal expressed with this construct didn't exhibit these exuberances. So we're now analyzing the samples to evaluate whether there is a phenotype in at the chiasm where slit is important. And I'll describe that a bit later. We don't have the complete answer yet, so I'd rather not tell you what we are not sure of yet. So to finish up, I'd like to acknowledge all the members of my lab, and in particular Sarah Baudet and Fiona Roche for still in the lab and were very important for this project, and Oriol Ross and Sandrine Coubet left the lab. They were at the start actually of this project. I'd like to thank as well all the members of the facilities of Institut La Vision, and in particular Eric Zagar, who was very important to validate that our construct targeted or excluded from lipid raft are actually going where we thought they would go. Thanks to our collaborators as well. I'd like to mention that we have a postdoc position available in the lab. So if you're interested in understanding how axons are, enter and behave in helping response to axon guidance cues, don't hesitate to contact me. Thanks to our sponsor as well, and to you for your attention. And I'd like to be I'd be happy to answer any question you would have.
All right, thank you very much, Xavier. This is a great talk, and you have here wonderful tools to really finely dissect second messages signaling. I think this is amazing work. Thank you very much for presenting this to us. And yeah, we have, so it's open for questions, and I see Philippe already is ready to ask a question. Philippe, to go ahead.
Yeah, I was wondering about as you know, the response of slit is dependent on the substrate. Huh? So what kind of substrate did you use actually in your experiments?
Yeah, so that's indeed a very good question. Yes, the response to slit, but to many other guidance cues is actually dependent on the substrate. Here we were on laminin, so polylysine and laminin. And unfortunately, we have not many choices for retinal ganglion cell axons. They are not very nice with us, and we didn't find any other substrates to try.
Okay, because you remember the story from Kim showing that was I believe it was fibrinogen. Huh? She used laminin on fibrinogen, and she had two different responses.
And I think it was related as well to different slit fragments.
Yeah. There was an internal part of slit on the C-terminal part, and the response was different as well. So, yeah. So that was I was wondering if, or maybe I'm wrong. Maybe the difference in substrate can induce a shift in a response from lipid rafts to non-lipid rafts.
There is for sure a tight interaction between integrins and lipid rafts, and in particular on the cyclases of integrins and lipid rafts.
Yeah.
So it is very possible that with laminin we're having we're selecting a specific response.
Okay.
I'm not saying that what we have here is the description of all the response downstream of given guidance Q. It might change depending on the substrate. For instance, the netrin response changes when you have laminin or when you don't have laminin, and it's related actually to the level of AMP. So it is very possible that changing the substrate, you change the localization of the response.
Yeah.
Now, what I think what we've shown here is rather that surprisingly, we have different compartments for different cues, and that the second messenger is not the place of the signaling pathway where the response is integrated, because we still have different response in different places, but at the end, the growth cone has to somehow integrate all the different cues that it detects to translate that into a change of morphology. And for quite a long time, we thought that, well, maybe second messenger is an easy way to do that. It's probably downstream.
Okay, thank you.
Thank you, Xavier. Next question from Alex Kolotkin.
Hi, great talk, really interesting. Can you speculate somewhat generally about what you think the implications are for two different repellents having this differential segregation in rafts? Like, for example, do you think the potential roles in regulating interstitial axon branching by slit, for instance, may be different from, let's say, negatively regulating terminal axon branching? Because it's interesting, I noticed you saw no axon retraction in the context of slit, but retraction and collapse. So in terms of thinking about sort of the relevance of this, what are your thoughts?
Yeah, absolutely. So that's something we are trying to investigate right now, actually. I would say that at least for retinal axons, ephrins, and slits are doing two different things. So slits are at the chiasm, the axons haven't completed their outgrowth. They still need to be guided, but not stopped in their outgrowth. And this is very different for ephrins. When they reach the ephrins target, they need to stop their outgrowth, start branching, and find their correct place in the target. So I would say that yes, there is maybe a stronger recursion with ephrin that would be a long-term effect on the axon. So just saying, okay, now you're at the correct place, stop there and move to the next step of your program. And with slits, the axon has actually not to do that. They just have to stay in the corridor and complete their outgrowth in the correct direction. So now it doesn't completely answer your question, but I would be very happy to understand what are the different morphological changes that are induced by slits on one hand and ephrins on another hand. So at the cellular level, what I just said is more in the system level. At the cellular level, I have no idea yet. I hope we're going to have some cues soon.
Great, thanks very much.
Thank you, Xavier. Jeremy has a question.
Thank you. It was really fascinating talk, and these are really powerful tools. I was wondering if you have thoughts about, I guess, following from where you were going in the last question, are these differences in second messenger signaling due to, I guess, intrinsic properties of membrane compartments, lipid rafts or non-lipid rafts, or is it due to differences in ligand receptor interactions? Like, is there a way that you could relocate or relocalize Robo or FA and then would that flip the responses, or would it not flip the responses? Like, would it stay with the rafts or stay with the receptor?
Yeah, that's very interesting actually, since for netrins, for instance, there is a switch of DCC from outside lipid raft to inside lipid raft upon netrin binding. Is this the same for robos and FAs? I have no idea. I would say that what we detect with the biosensor is really a short-term response. So that might be, so it's let's say, well, medium term, like minutes, not seconds, but minutes. So I would rather say that this is the initial response. Now, there is a possibility that you have a second step of the response that is induced after translocation from outside to inside lipid raft, or from inside to outside. Now, is that related to the initial localization of FAs and robos? I really tried hard to find a description of the subcellular localization of FAs. There are many for ephrins, surprisingly, but not for FAs. So I don't know if they are in lipid raft, if they are residents of lipid raft or not. I'd love to do that. I'd love to know that. But for now, I have no clue of where they are. I would guess they are in lipid raft, and that would be the same as what is usually reported in reviews, just generalizing the description of the localization of the ligand. But now, is it possible that these second messenger signals are the ones that translate, that instruct the translocation from one compartment to the other? Why not?
Okay.
But I have no data of supporting that.
Okay, thank you.
Thank you, Xavier. There was a question in the chat from Arthur Kanaya, so just before he left. So he asked whether you have any ideas about ephren B signaling in this particular context. I was about to ask this one too, but so.
No, I can't say much more than that. No, we haven't tried other guidance molecules. We designed the project set to find the integration point. We failed and found something else. It was very interesting. But so that's why we focus mostly on the regular, well, some of the regular repellent molecules, and didn't really check ephren. I would say as well that it would be very interesting to have an overview of all guidance molecules, but this is a massive amount of work, well, I'm not sure I'm going to be able to convince Sarah to do the experiment.